big data person, generally speaking. But I want to start off by reminding you all that math is A, not scary, and B, you've been doing it all along, and C, you're actually in a program that is particularly equipped to think about big data in a way that not most of the PhD programs here are. And it's because if you think about how genetics was studied, going back to Galton, like, it's really about modeling these behaviors, sometimes even when we don't know what's causing them. We can't necessarily see them or see it directly. And then using that to make a prediction. So how can you find the structure and the behavior of a system? And that's what the mathematics really does, more than anything else. It takes these behaviors and it turns them into something that you can then operate on and allow you to do prediction. So what do I mean by unseen structure or unseen behaviors? So let's go around and if everyone can give me the first letter of their name and then what lab they're in, what building they're in, and what lab they're in. So go ahead.
The first letter of the name is L. I'm in CMSC in the GFC lab.
Okay.
First letter is V. I'm MRV, and I'm in the
Okay.
J, MRV, faculty.
H, MRB, party.
MRB,
Z, MRB, and
Okay.
A, Rango's S. C, CRB2, Belgilescu.
Okay.
Another A, Rangus. Fifth K.
Okay.
D, Rango's U.
You
D,
guys are the.
No, no.
Okay. Sorry.
I'm D, MRB, Lee Lab.
Okay. So we were going to cluster, right? Which is like the first thing that most people do in a high dimensional space, right? We were going to cluster based off of the letter in your name. Would we find any patterns? And would they be meaningful? So I probably should have written them down. Normally I have some things in front of you. I should have written them down, but I'm pretty sure the answer is no. The distribution of letters is pretty random here. Now, we didn't have any overlapping labs. That happens generally later on. But if we were to cluster based on location of where you're currently rotating, would we have any structure? Yes. We've got a Rango's cluster right here. We've got a MRB cluster kind of here, right? All right, so we have this structure that's starting to emerge. What property is that structure reflecting?
Probably the feature that is most closely related to it is the distribution of faculty in our department.
That's a good one, yeah. What other property might it also, if we get a little bit deeper, so if we wanted something that said about the relationship of the students who are causing the clustering, what else might that be?
Interest area.
That's a good one. What other types of structure might there be? It's a very basic psychological thing. Exactly. You're more likely to sit next to the person who you see more often, right? It just happens to be, right? So there are these behaviors of people, of the department, that come out in this very physical property that we can measure. And that is, again, what, Mendel observed, what Galton observed. They did this on very gross phenotypic levels. And what has happened is that now the molecular biologists caught up. And now we can take these phenotypes and measure them on the molecular scale, right? So now we can take these traits and we can start to see what can we look at in a cell, in the genes, in how those genes are interacting together that really drives information, that's creating the phenotypes that we're interested in. What other structure can we observe? And so, we do this, at least in my lab, by measuring gene expression on the cellular level, right? Because cells are fundamentally a unit of life. So if we treat each cell as a person in class, where they are clustering, where they are existing, and where they are behaving, telling us about the data. And so one of the fundamental ways to do this is via dimension reduction. What does that mean? So it means that you take the 22,000 genes that you have measured in an individual cell, and you look for different dimensions that can describe what that cell is doing. And there are ways to do this mathematically, and I'm going to walk you through 3 different ones. But if we were just talking about people, what are potential dimensions that you might measure on? We can jump to cells next. Yeah, exactly, right? Just fundamental properties, right? So if we're looking in gene expression space, what do you think these dimensions come out to often be? What are things that genes work together to do in a cell?
I guess the first answer would be like cell type, but specifically like the functions of that cell.
Okay, so how do you define a cell type?
Firstly, you could, in a circular way, you could say it's the cells that look like each other.
Oh, really? That's very interesting. Okay, so there's 2 things that I will comment on there. The first being that the transcriptional profile of the cell and its morphology are not necessarily well correlated. So that's something to be acutely aware of. The second thing is, okay, so if you're talking about morphology, what molecular programs are driving morphology? Cytoskeleton, actin, your different, these are not my areas of expertise, but things that other people have talked about in this class. Okay, so you would expect to see similarities in those programs, correct? Right? And maybe if you take those 2 cells whose gene expression profiles are not well correlated, but who look very similar, and you just subset to the molecular programs that drive cytoskeleton, actin morphology, those programs will be similar, right? So now you're taking your 22,000 genes and you're subsetting them to just those that are involved in those particular processes. All right, what other things might be important? And this gets back to cell state versus cell type. And one of the things that comes into question when we start thinking about how are we going to measure these things, how are we going to analyze them. So what about cell cycle, right? So cell cycle is almost always one of the primary sources of variation in a system. If you have dividing cells, their transcriptional profile will vary depending upon where they are in cell cycle even if they are the quote-unquote same cell type. And because of this, for many years, people have regressed out or taken out cell cycle information. But the rate that cell cycle occurs, the length it takes, and where you are primary drivers in fate specification and development. They play a huge role in a lot of different diseases. So what information are you losing by regressing out that information, right? Are there ways to capture it? That is something we do in the lab. All right. What other dimensions might there be? This also gets into the idea of linear versus non-linear programs. So something like cell cycle is a cycle. It's an inherently non-linear process. So when you use analytical methods like regression, like many forms of clustering, like many forms of dimension reduction, like principal component analysis, these are all linear methods. And so they have to be adjusted for in order to account for non-linear effects. But there are also ways to do that. It's one of the reasons people have been pushing for AI and neural nets is that those are inherently non-linear analytical methods. Now, in that non-linearity creates instability. So it's not necessarily you're just going to put it into ChatGPT and it's going to solve it for you. I assure you, it gets a lot more complicated than that, but there are opportunities there as well. All right. So why don't I take a step back and give you a pretty basic example of a set of linear methods, right? And we're going to work in 2 dimensions just to sort of show how you can potentially do this. So what I'm going to do is I'm going to take 2 genes, in this case, I always forget to check which ones I'm doing. Let's go with SOX2 and PAC6. And if I'm wrong about the biology here, I apologize in advance. Right? So I have the genes expression, right? So I'm going to plot a whole bunch of cells. All right? So each one of these dots is a cell. Okay? And I'm going to put a little bit more density here, just because we'll do clustering too. Sure, why not? Now, if this is single cell gene expression data, right? You can think of this as your high-dimensional UMAP reduced down. One of the first things most people will do, and I'm sure most of the papers you have read, will do is clustering. And like I said, there can be information in clustering. But when you're looking at something like this, you have to be acutely aware of the fact that there are multiple input arguments to clustering algorithms, but the fundamental input argument to a clustering algorithm is the number of clusters you are looking for. So if you ask for 1 cluster, it'll give you the whole data. If you ask for 2, it'll probably do that, right? But the degree to which those are actually 2 separate clusters is highly variable. And I would say if you did a silhouette scroll, it probably wouldn't be great, but most people don't actually check those. They just say, hey, look, I got 2 clusters. Maybe they're going for that new cell type in the Nature paper, so they put in 3, and they get this, there we go. I just identified a new cell type. Woohoo!
It's specific to disease.
It is. I mean, it happens to be 1 cell, but that's part of it. It's very
From 1 mouse.
Yeah. So there you go. And you will get trends in this, right? Because there is a difference between this population and this population. But this is not necessarily the most informative way to look at this, right? Let's do something very basic. Let's do a regression, right? And actually, in the 2-dimensional case, a regression and a principal component analysis are very mathematically similar, and I'm going to show you exactly how, right? Because remember, principal component analysis teaches you the axes of greatest variation. So what does that mean? It is greatest variation is the greatest amount of change, right? Variation in the system, how one person is different from the other. Now, it conflates a lot of things, and we can get to that later. But first, let's just do it. So, what we do is we look for what is the greatest direction of change. In this case, right? It's just like this is a regression line, same as the principal component. Right? And so what you can see here is, and this is important in terms of thinking about genes and how they're actually working in the body, because they're not working in isolation. In the same way, height and weight aren't independent of one another. There's correlation structure, right? They work together. The question is, how do you break that down into what part of this gene is contributing what portion of its behavior to each part of the thing, that molecular process you're interested in, and then how do you associate that? All right, so in that case, this is PC1. This is, if we were doing linear regression, right? Your gene expression in this case would be SOX2, and your this, right, is the change in SOX2 based off of the change in PAC6, right? So just, okay. Now, principal components are orthogonal. In regression, you're then looking at the difference in the residuals, right? The change that is not accounted for by this slope. So if I take one step here, right? Let's do this guy. I can move this much along. Now if I want to account for the difference between the value in the line, sorry, this is where PCA and regression are different. And the difference in this value and the line, right? This is your residual. It's your residual variance in regression analysis. This is why residual variance is important to think about and account for. It can actually be the error can be the place that's the most interesting. So if you regress out cell cycle, and this line represents the variance that's accounted for by cell cycle, all the information that you have left to do analysis in is here. That's it. So when people do GWAS studies, and they say, we take out the first 10 PCs and look at what's left over, all of that they're studying, that's it. That's all the information they have left. They've taken out everything that's accounted for by this entire dimension, right? That information is gone. All right, so again, in regression, it would look like this. The difference between PCA and regression is the fact that regression is supervised. What does that mean? It means I told the algorithm what X was and what Y was, right? I told it I'm looking for SOX2 and I want to model SOX2 based off of PAC6. PCA doesn't know any of that, it just looks at the dimensions. And then the other thing that the result that you have to think about in terms of how that changes things, right, is that PCA makes the PCs orthogonal. So now, because I'm not looking to keep everything in SOX2 space, I'm looking at the dimensionality itself. I'm actually shifting the data or rotating it. Sometimes you'll see in the results of the algorithm, they're called the rotations, PCA rotations. What you look for, and this is me actually not being totally accurate, so the amount of information that people in GWAS are looking at is actually this. It's relative to the line, okay? And this rotation, by the way, is the rotation that they're talking about in PCA algorithms. So, all right. All right, so then in this you would have, we'll call this PC1 and PC2. And remember, this is a rotation. So, now, like I said, you can do this clustering, you can get your continuous clusters, and you're going to get information because we know that there's structure there, and we can capture this structure in these PCs, right? So, or in these beta values. It's the same thing. Because you're solving an equation that's extracting information. And in this case, you're solving based off of, right, the gene expression of the cell. So now, what we can do is say, what do these continuous dimensions correlate to? And this is actually something that I have had a lot of experience with in terms of development, but also in terms of some disease processes. Sometimes this is your disease, right? Your severity. And it's low here and high here. In the case of PAC6 and SOX2, what ends up happening is actually this quadrant are your progenitor populations. And once you go from negative to positive in PC1, you go from being a progenitor to being a mature neuron. That's the transition. And so this gradient then actually represents the continuous gradient in the developmental process that goes from progenitor to neuron.
In looking at like this plot in isolation, how do you know if it goes from progenitor to like final differentiation and not going the other way?
Metadata.
Okay.
Metadata or the loadings of the genes, right? So you could look in the loadings. So you know that genes that have above this expression of these two markers are, this type of it's marker gene. But you can do it in different ways. And the thing is, if this was the full dimensional space, right, you don't actually have to rely solely on marker genes because you'll have values. This is the benefit of continuous processes. You will have a value for every single gene. So people call PCA dimension reduction. It's actually not. It's a way to reorient such that you load the most information in the higher dimensions. But every single gene will have a value in every single PC. That value may be 0. That 0 is informative. If you do a clustering analysis, you will often not get that to be the case. And you'll definitely not get it for every cell. Not that there isn't a time and place for clustering. I don't mean to hate on clustering, it's just overused. Okay, so negative versus positive is progenitor versus neuron. So then what's captured by PC2? What is this giving you? Well, in this case, it is, in the eye, these are amacrines, and these are bipolar cells, right? And you know this again from the annotation. You can get that from looking at the cells. You can get that from marker genes. You can get that from, you can make that call in the space. And what's interesting and you now have the ability to look at is in this space, if you had done, so first of all, you would have called them all the same cell type to begin with anyway. They'd all be progenitors, right? But in this space, you can start to see that what's guiding towards an amacrine or towards a bipolar cell is these lineage markers, but the difference here is not large enough to be statistically significant. It isn't until you get into the neuron that it is. And this is a way you can start pulling out some of these dynamics, start really getting into processes in a way that you can't necessarily with just like t-test style analyses. And the way that you're dissecting this data can really be biased in terms of like, well, maybe you want to add additional constraints. So right here, we have negative and positive. And we're able to make inference on that because we have information about the cells themselves. We can correlate it with, oh, we know these are progenitors. Or we can correlate it with a process that we know is related to maturation. What if you didn't have that information? And also, what if the idea of a negative value in gene expression space isn't inherently intuitive, right? Tell me what negative gene expression means. Because I'm pretty sure that in the cell you can have 0 gene expression, and you can have, I mean, someone once argued with me, well, I don't know, it's a microRNA, it's soaking it up. But like, really, even if that's true, it is two positive things that are coming together, right? There's no negative expression.
You can't go below 0.
You cannot go below 0. So maybe you want to put a constraint on how these dimensions are learned. So the way to do that, again, something that we do in our lab, and this is what the paper that you guys had to, I forced you to read was on, is called non-negative matrix factorization. So PC1 and 2 is a matrix factorization. You take a matrix, right? Your genes, 5 cells. This is your data matrix, your Excel spreadsheet if you want. But these are the things that you've measured, right? You physically measured them. And you can decompose them into 2 matrices. One that is genes. I'm going to call these latents. And 2, I'm calling them latents for a reason. That is cells.
This one works.
I think it got wet. There was somebody sprayed and then wiped. That is cells by latents. I can't spell. All right. And this, by the way, is this. It is your PC by loadings, right? It is, all of these equations are all the same. So yeah, your latents can be PCs, they can be your betas. In this case, right, this would be your betas, and this would be your X values, or your design matrix for your regression. because the design matrix would be based off of the metadata that you have for your experiment, right? The way that you actually write that is, you guys have had Casper, right? No? Casper?
No, he doesn't teach.
All right. Well, all right, we can do a more in-depth progression another time. We're going to get into NMF. So, say you have, say your cells looks like this. I'm going to do this. I'm going to make this one like that. We're going to solve this a little bit backwards. Right. I'm going to stick this guy at the end. I'm sorry. I should have planned this better. So we'll start here. Okay? So if we're going to break this into its components, and this is where you actually get into dimension reduction with NMF, I'm going to ask for 2 dimensions. And in this case, I'm going to ask for 2 dimensions because I'm looking at this and I'm like, that looks like there are 2 temporal processes that are going on. One that's induced early and one that's induced late. So those are the 2 processes, right? All right. So these are my 2 latents. These are my 2 latent spaces. These are the 2 dimensions that I think are capturing the biology. This matrix right here. So how do we, what we're doing then is we're solving for the genes now, right? We're going to optimize over genes. So let's start with this one, right? Here we'll do A, B, C, D. All right, so we're going to solve for A, right? Because that's what this is. It's genes by latents. Okay? So this is latent 1, and this is latent 2. Okay? 1 and 2. So start with an easy one. Gene B uses all of latent A, right? So 1. And none of latent B, right? So that's the value for gene B. And so you can go down, you can see gene B uses 1 here, none of 2. All right, let's do gene C. It's the opposite. Uses none of latent 1, all of latent 2. Gene D, 1 and 1. All right, who wants to do A?
1 and 2.
Very good. Exactly. So it uses 1 of latent 1. And just to show that this is not a binary process, and that this is much higher, right? 2 of latent 2. This is our solution. This is that. And so you can see now how you're able to extract gene usage from this type of decomposition, right? This is just matrix decomposition. The constraint we've put on it is that the solution can't be negative. That being said, like, you don't necessarily see that here, but you can see, right? If you go down this column, it gives you all the genes that are involved in this early induction. This is what you do gene set enrichment on, right? That gives you your, what is this pathway, if you don't know? Or what are genes that are coming up? Maybe you didn't know D was there, right? Because it was hiding before.
So in that case, when you put like gene enrichment, like you put it, just like ORA, overrepresentation, just if you put C there, gene C,
Yeah,
it will, in that pattern,
yeah,
C is the value, the knowledge regarding C is that C is not there.
correct.
However, when you do gene enrichment, it's not necessarily, people might, because C is now in the list.
Yes.
If
So
the
that'll
enrichment
count against
pathways
the
that are like
enrichment.
really not enriched, is the opposite of enrichment, it's depletion.
Then you need to do a different type of enrichment test,
Right,
but which you can do.
okay.
Yeah.
Maybe we can talk about that.
Yeah, no, actually. But the other interesting thing that you can do, and we'll get to this, we're getting there next. Go ahead.
So, as I understand it, you have some genes, you define some number of latents, and then you're basically doing some sort of like gradient descent to get as close to the original matrix as you.
If you are doing a VAE, which is different, and I guess, yeah, you can do a gradient, just like,
I mean, I don't know what
square.
I don't know what the technical term is, but
Yeah.
you're doing some process that gradually moves, your latents to some point to where the multiplication of those two gets as close as you can to the original
Yes,
matrix, right?
correct.
How do you decide how many latents to use?
that is a fundamentally unsolved mathematical question, but also something we work on in lab, because it's cool. And it's cool because biology is hierarchy. So the answer is, what question are you asking? Are you trying to distinguish blood from the brain? Because that doesn't take as many latents as trying to distinguish, cortical neuron layer 2 from cortical neuron layer 5, which also actually does not take as many latents as trying to then distinguish cortical layer 2 from cortical layer in the auditory cortex from cortical layer 2 in the somatosensory cortex. Because even though those do entirely different things, well, I mean, they're neurons in the brain, but like auditory versus somatosensory, it's a little bit different. They're transcriptionally more similar than different layers in the cortex in the same area. So what does that mean in terms of information content, in terms of functionality, in terms of the fact that cells that look similar often are transcriptionally identical, they're different, right? Why is that? What is the functional source of variation? And in terms of trying to figure out how many latents, basically what are you trying to disentangle? And if you can't break it out into, what does that tell you about the primary sources of variation, right? The reason that they take out the first 10 PCs in GWAS is because though that is driven by, and I should not be lecturing about GWAS because it's not my thing, so if I'm wrong. So the reason that people regress out cell cycle, right, is because it's this primary source of variation. It gives you a huge amount, but if that's not necessarily exactly what you're looking for, if that's not what you're interested in, then if you want to dig into that little bit of residual variance that's left, if that's where your signal is, then that's where your signal is. But that's also why computational analyses are not magic. And if you take anything from this class, it's that at the end of the day, even if you just work with someone to do your analysis, know every decision they made. Because the answer depends on it. You do not put something into a computer and hit go. It will give you the defaults, and you better hope that someone tuned those defaults well to give you some sort of nonsense, because every decision you make in an analysis has an effect.
So kind of bouncing off that question, you said that the number of latents you use is like unsolved, but my question is, how do you ensure that like you're not either like underfitting or overfitting like your
Does it replicate? Does it generalize where it should?
So like if you were to use another data set, for example.
So, the best part about all of this, these lovely vectors can be fed back into this algorithm and solved for. It's called projection. These represent dimensions. Latent spaces are dimensions. So just like what we had here was actually a two-dimensional space, right? These dimensions were defined by Pack 6 and SOX 2. And this is why we started this whole thing by my asking you, what are the dimensions that define people? What are the dimensions that define, where you're sitting in the room? What are the dimensions that you're actually solving for, right? So here, these are dimensions, right? PC1 and PC2. We just solved for them. Here's the values. We can take these, and remember how I said this is called a rotation? This is an angle. And we rotate it so that now, instead of plotting everything relative to Pack 6 and SOX 2, we can plot relative to PC1 and PC2, or latent 1 and latent 2, NMF1, NMF2. Your coefficients in your regression now become your design matrix. So the gene expression signature that you learned when you regressed, disease versus control is just this, right? Where this is disease and this is control. And you have two different signatures, right? Same thing. And now, to get back to the Pack 6, this analysis, right? We take data, in this case single cells. We plot it in here. I have never seen this data. This data doesn't have any labels as far as I know. I can tell you this cell is a progenitor that's fated to be a bipolar cell. This cell is a neuron that is fated, or that is a bipolar cell, based off of where it exists in this space. If I trust my bases, if I don't trust my bases, if I don't know if the latents that I learned are right, I take a data set where I know that these are bipolar cells, and I know that these are amacrine cells, and I know that these are progenitors, and I plot that data into my bases, and I say, what score does it give me? And this is a huge warning. So like Allen Institute has a algorithm right now. It's literally called Map My Cells. And it's based off of further proof that clustering is not the way to do this. It's based off of clustering and doing essentially this type of calculation. And it will tell you that there are gut cells in human hippocampus. So like benchmark, try multiple algorithms, and then also like the question, what is the question? Are you checking the latents? Are you checking the algorithm? And trust nothing.
We thought we had found a very interesting pattern. I was trying to find a pattern that would identify cells of a certain origin, embryonic origin in the adult mouse, so I didn't have to trace them anymore.
Yeah.
And we were like, oh my God, this pattern was picking up like the green cells that we have traced.
Yeah.
And we were so excited. So I was like, well, let's see. Now let's plot this pattern into a piece of the aorta where these cells should not exist. And the pattern was still there, even though maybe it was like, oh, the cells were sorted on a Tuesday pattern.
Well, I mean, no, and that's the thing though, that's the problem is if these are also in your tissue of origin, there will be signal, right? There are genes that are shared between the gut and the brain. There's a lot. I have a whole grant on looking at a program that's used actually in the heart to separate first heart field from secondary heart field that also then shows up in the eye to separate up retinal ganglion cells that sense up motion from down motion. And the reason that this program it's very similar and it seems to coexist is it takes cells who are very transcriptionally similar but need to be functionally distinct in terms of their spatial location, and it drives the separation separation in space. And it shows up in the heart and it shows up in the eye. And there's a couple of genes that are swapped out in terms of tissue specificity, but the program is strongly conserved. So it happens, but also you need, if you're just using it to label, right? If you're blindly labeling, don't label gut.
Now in this case, what we wanted could not be used for the purpose we wanted. Maybe there is another purpose for which it's okay.
Yeah. You just, again, what is the question? What is the level of resolution that you need to answer that question?
Hopefully this makes sense as a question, but like for my understanding, like for the disease, like you kind of have to maybe assume that it's the same, or a portion of it is perhaps like maybe the same for the population one is like a control cell and like a disease population. But if there's something like an inherently different enough about like the disease population, like would your latencies transfer as well, or would you have like the same variation?
No, they won't. They'll fail to transfer. And actually, well, it depends on how you're doing it. So with the transfer learning algorithm in our lab, we condition on the latents. We condition that you've done your due diligence and you know what your latents are and you trust what they are. Because in my experience, by the time they're well defined, by the time you've done the experiment, generated the data, analyzed the data to the point that you have signatures, like you really know what they are. And so we condition on those, rather than these like global sort of categorizations. So if you can condition on those and you use the type of transfer learning we do, what's really cool is that if that signature, if that structure isn't there, it won't show up. So you can get things, you're more likely to get false positives than you are to get false negatives, which is potentially really powerful. And we've used that, so it's unlikely that it was a batch effect, but it is likely that it's a biological process that's more global or just not super well defined. Technical artifacts tend to be very discrete and don't transfer well.
Yeah, I don't know. I mean, maybe it was apparent. In the end, we don't need it anymore, but it was like, it was picking up all the cells. It was not GFP, but it was correlating with the GFP.
And that's another good point, right? Like the question then becomes like, you really need to design your experiment to have that metadata to check. You got to ground.
So just like kind of big picture here, you might do this analysis, you learn a set number of latents which might might represent some biological program or like metagene kind of thing, and then you assume that the programs are being used in another part of the body, just to different extents. So you take essentially your basis, that first one, and you're just changing the coefficients of the second one. You're saying the same programs are at work here, they're just being used to different amounts, so we're changing the coefficients? Is
You are
that-
not changing the coefficients. What you are doing is you are solving for the usage of that latent in those cells, which is how you're making inference, right? So the coefficients, right? So if you do a regression, all right, I'll teach you a regression real fast. And then I can put this in autoencoder land if you really want me to. But you lose a dimension, which makes it harder to make inference. You end up having to do a lot of correlations, which is not the greatest. Also, they're not super stable. Okay, so so these are always genes by latents, right? And this is always latents by cells. Remember that. These have to be this way. The math does not work otherwise.
But are you changing the value, you're just changing the values in the second matrix?
So if you learn these,
Yeah.
right,
Mhm.
then, and this is your data, right? So you learn these in data 1. You make inference based off of your cells. Keep this constant. Data 2, right? B stays the same. Solve for X. What is X? Latents by cells. This cell uses it, this cell doesn't. This cell uses it, this cell doesn't. This cell uses it a whole lot.
How do you initialize when you're learning for data 1? How are you initializing your latent space?
Depends on the method. So that is a method dependent thing. So if you do PCA, it is an empirical solution. It's literally just an equation. You solve it. There's a little bit of estimation based off of the fact that these are not beautiful, symmetric, semi-defined L2 spaces, but you can get them there. It's part of the reason you do a log transform. Be aware of your pre-processing, it makes a difference. If you do NMF, depends if you do gradient descent versus MCMC, same thing. Most of it's based off of singular valley decomposition. If you do autoencoders, speaking of initialization, you have your data. Right? You have your data. You take your data, you encode it. By the way, this is just a lot of math, with the exception of each time you do the matrix decomposition, you take the product of that matrix. That's not even a decomposition, it's a dot product, and you put it into an element-wise ReLU function. You get your latents. Notice a key thing about these latents. They're very different. What is missing in these latents? What are you missing? It's important if you want to make inference. And then you get your reconstructed data, which is not to size well. Oh, come on, someone give me a good guess. I kept my dimensions fairly similar.
You don't have the genes in there?
Bingo. Neural nets can only compress in one dimension. In order to recover the other one, you have to do a post hoc transformation. So no matter what, if it's an autoencoder, if it's a large language model, if it's a transformer, almost every single one of them can only compress in one dimension. So you will only be able to learn the information that way. So you need to make sure that if you're choosing genes or cells, now you can recover it. There's ways around it. But be aware. Yes, this is cells by latents. And then what you do here is, so you compress the data, you re-expand the data, and then you compare your original data to the recompose data, and you optimize that way. And again, the constraints in which you do so, it depends upon the loss function that you define for your VAE, or for your encoder, variational autoencoder being a type of autoencoder. Transformers are a little bit more complicated, but you really want to go into those. I feel like we're straying farther and farther from the biology. If you really want to go into transformers, come rotate. You've got a project. It's actually very cool. The attention mechanism, while debated about hotly, is
So sorry, I'm like selfishly asking all the questions.
yeah.
So in the when you ask, say, the genes by latents, you get. I know I read the paper. You get a rank, and you say, oh, you can. You know, their rank. What's their rank in there? What does it actually come from?
So well, it depends. So first of all, are big values good or small values good? Because again, this depends on how you've written the algorithm. So let's go back to our 2101, right? So if I were to rank on this, right, I would, so we'll
So
do
A, B, C, D. We have.
yeah, disease and control, and A, B, C, D.
But in this case, 1 and 2 are, patterns.
Yeah.
Yeah.
Yeah, no, so disease and control, right?
Okay.
Yeah. No, these are the weights,
Right, sorry.
right?
Mhm.
So for C, in terms of, or for your disease pattern, in terms of ranks, if you're just ranking within this column, you're screwed because you got a bunch of ties. So there's no ranking here, it's arbitrary. The only thing that's useful is not B. For C, you would, A would be ranked highest, right? But, and this is something that comes down, actually brings out a very interesting question, right? So A is ranked highest because the expression value is higher. But if I wanted a marker gene to go back into my dish for the control, or for C, right? A is not actually a good one.
Yeah, B is better.
B is better. So I don't think it's in this one, but so like during my PhD, we actually defined a statistic to find the genes that are more uniquely associated with latents as opposed to the highest ranking ones, because if you just look at weights, often, because right, you have a high level of expression that you have to parse out, your weights are often biased by the fundamental gene expression level. So sometimes you want something that's more specific.
So, but broadly, the law, if you say whatever, for this pattern, unknown, pattern 2, we don't know what it is. And
Right.
there is a top gene, and then they're all ranked. You can do like a GSCA thing.
Yeah,
The
for
bottom,
each
the
GSCA.
ranking is from there, and the top is broadly associated with more expressed and less expressed.
Yes,
Okay.
often. Yeah, often.
I thought I didn't quite had, because sometimes what's not expressed is also informative.
It's very informative.
So I was like,
It's actually, yeah, it's very informative. And when you do projection, that's one of the things that blows my mind. You need the zeros. You can't just do the top 100 genes if you really want to resolve anything. What's not there is often as, if not more important than what's there. All right, did I leave enough time for discussion? Should I keep
Enough time for
going?
one discussion.
What's the plan, man? Looking at you, Kirby. I can keep going. You know me.
We had 2 presenters, so they were going to get 30 minutes apiece. We have 30 minutes left. So I was presenting on
I think that you should, it's up to you, I think you should finish up and we'll work out the
Yeah.
discussion.
Yeah, we'll work out another.
You sure? I mean, especially if someone's going to present on the paper I sent, like, you can hop in. This is like the bulk of what I was going to talk about. So, like, I think, yeah. Go ahead. I can haggle. I can stay and haggle if you want. Whoever's got my paper. Because it's pretty much this. It's the decomposing cell identity, right? That's the whole idea. How do you decompose cell identity? What's relevant? What are the different?
Can you call latent matrices tensors in this case? And are you plotting by tensor?
So yeah, it's just a tensors. So not exactly. And here's why. A matrix is two-dimensional. A tensor is a multi-dimensional matrix.
Right.
So yes, the question then just becomes, how do you scale the math to the additional dimensions? Now, the interesting thing there is that it depends on what you want to do. If you're asking because you're using PyTorch and you're just like trying to figure out how to reason in terms of tensors versus like matrices, it's pretty much the same, don't worry about it. If you're asking because you want to do a tensor decomposition where you're taking in another dimension of information, that requires a expansion on the math that's a little bit different, but it's fundamentally not. It's just, it depends on what you want to do. Like, it's not a big deal. Essentially, yes. There are tensor methods for most of the things that I talked about. Yeah. It's literally just like, okay. So if you were doing, right, if you had your cells by genes, that's a matrix. If you wanted to add into this space, into the data space, not the solution space, the data space, additional level of information, like disease versus control, or height, or something where every single element has a value, right? So in cells by gene space, it becomes a little bit more complicated.
Could be
for
like
people.
the spatial location.
A spatial location, there you go. Perfect. That's a great example, actually. That's the one that we're doing. Yes, I should have thought of that, right? So if you wanted to add that dimension, well, I'm not doing tensors, right? On this side, not on the solution side. So not on the side where you're trying to interpret, use that as an interpretation, but as something that's in the data side. That is a tensor, right? Because you're solving for a cube as opposed to a square, right? Your data, you just scale your data significantly, right? Each one of those is well, it's not a cube. Well, it is I forget what the word for a 3D rectangle is, but that's what it is.
So you generate the latent matrices from the tensor. It would still be, I guess, a 2D matrix or an XY matrix.
No, you get multiple matrix solutions. That's why.
Okay.
Right? It becomes a little bit more tricky to deal with. So you get multiple matrix solutions, but the reason that this is hard is that you have to account for this degree of variation and these interactions and these interactions, right? So you're solving for both a cell by space, a gene by space, a gene by cell, and I think that's it. I think that's all. gene by cell, gene by space, space by gene, yeah. So you get three matrices, right? All of which have coefficients that you then need to reason about. And you're looking at the interactions of all of those, which are important. And so in the spatial context, it's actually really important because space creates autocorrelation, right? People who know each other are more likely to sit together, right? So if there was something else that was associated with that as well, right? Like what if you all had A's, names? Then you might be like, oh, it's the A. And then that's like you put a lot of that's super significant, right? But actually, there's a spatial component that's overinflating your significance for something that's not being accounted for. You see this a lot in ecology models and disease epidemiology studies, like the prevalence of disease and how it spreads and different things like that. But yeah, it's starting to be brought in. The stuff we do, but we didn't have the ability to really look at space until now, so. Or at least not on this scale.
I'm going back to the beginning of the first week, and we were assigning like X and Y. Is it just you're choosing like two variables at that point, or like if you're trying to account for like the entire process, how do you?
So yes, so for regression, you always choose X and Y. Always. The way that things like monocle or Seurat scale to doing single cell is the same way people scaled with GWAS, in terms of they just put a shit ton of compute behind it and solve that regression of a single gene's expression versus the model 22,000 times, right? That's still what's the going size of the genome these days? That was, when I studied for quals, it was 22,000. I will forever remember that in my mind. And I'm like, I should probably check to make sure that that's correct. But, because I don't actually check what we align to these days. Because there's a lot of pseudogenes in there, so, in the on-code when you do alignment. I don't know if they count. But, so yes, when you do regression, when you do a singular value decomposition, you're actually decomposing the matrix and solving for it, so you don't have to assign an X and Y. The equation does it for you, and you're learning the structure of the data. And what you're doing is basically saying, I want to learn a particular structure. And that's basically, so the shape of the latents, or the constraints that you put on your latents, can they not be negative? Do they look like Gaussians? So generative, additive, generalized GAMs. GAMs, for the most part, are based off of the sum of different Gaussian processes. Anyway, it's the shape and the constraint of what you're doing on that. Because you have to constrain it somehow. But that's going to affect the result that you get. So you just have to think about that, right? Not everything looks like a Gaussian. It's useful, but it doesn't mean that it's a good fit. And also, if you're ever in doubt, like, it's me, and Casper's a good person, like, there are people biased to us. Talk to people. Careful what you ask Don. He's convinced that you can solve marriage through PCA.
Marriage?
Yes.
do you need to cut?
Who to marry via PCA? I was like, no. So, but there are people in the department who do this stuff and who want to go. Just, yeah. Okay, so you want me to talk for another 15 minutes, or you, or do you really want me to talk for 15 minutes?
And so we don't have much time for discussions.
Sorry.
But if anyone has.
It's you or no one.
Oh, it's me or no one, okay.
You're the star of the show, so.
Jesus Christ,
go ahead and
you
cue
touch
that
it.
You can also just say you're done. Some of the teachers do that.
Well, what would be helpful? All right, is there anything you're like, would like to know? Do you want the regression lesson? Have you learned about t-tests and what that actually means? If I asked you what a p-value is, how many of you could actually answer and give me like a quals answer? Because that's my go-to quals question.
Ask me that on my calls.
Yeah, I know, I did. You did very well. So,
Was I there too?
I don't mind them.
yeah. All right, let's go over what a p-value is. All right, so p-value is the probability of observing a value as or more extreme than what you observe, right? And so the reason this is important is based off of what distribution, right? Same thing as your latents. What's the shape of that? So we start, right? You're doing the test. Most t-tests are called t-tests because they're t-distributed, right? What is a t-distribution? T-distribution is a normal distribution. Looks like this, right? Fundamental distribution. Right. Your t-distribution has fatter tails, right? Which means you have a little bit more weight at the extremes. And this is useful because it makes it harder to be more significant. It gives you a little, right? What a distribution is it's a density function. A density function is a count of how often something happens, right? So if we went around the room and we started with letters, right? We can make a density function for the distribution of first letters in this class. Does that make sense? And you could use that and say, as a null distribution, or you could use that and say, if I compare that distribution to the distribution of all of the different classes in the School of Medicine, what does that look like? And that would potentially give you demographic information, right? Based upon also age information. Some names are more popular than others at different times, right?
Although the classes seem to.
Well, there, you would have to control for size.
No, also like the time frame of where the name change may occur.
Yes, yeah. You'd have to look over it, right? So it's just a density. So if you look at this density, what is the most common answer? What is the most common value in this density? And we've normalized it in this normal density. What's the most common value? Yeah, where is the peak? Or if I was a good at drawing on the board anymore, which I'm not, I used to be.
0.
Yeah, it means 100. So the most common value in a normal the score function, say, is 0, right? When you mean center, you literally take the mean. The mean is the most common value, right? It is, or close to it, if it's a discrete distribution. Right? So the fact that there's more values here in a t-test than there is in a normal gives you a little bit extra weight. So literally, when you take a p-value, it's just like I go to that value, I see that's the value, I take the area under this distribution, and I look at, and that gives me the probability of observing that value. And so what is the probability of observing this value or anything more extreme? So is it a lot or a little? Little. Is it a lot or a little? It's more, right? So that's all it is. It's just the probability of observing that value or any more extreme value.
So in this case, this is one tail.
This is one tail, correct. So what is a two tail?
You're considering probably
Yep. You go on both sides. Because it's a symmetric distribution. What if your distribution looks like this? Speaking of single cell data. Woo! Did you do a two-tailed distribution there?
This is an elbow plot.
No, this is negative binomial.
Oh.
This is another type of distribution, right? So single cell data is distributed like this. Most of your genes are not expressed or very lowly expressed, 1 or 2 values. Very few genes are highly expressed.
I have a question about that.
Yeah.
So, where it's from the differential expression analysis based on knowing that that's a distribution, and it's kind of a problem because
Yes, it's a huge problem. You're doing a t-test on that. You think it's going to work real well? Not
So-
unless you transform the data a whole lot.
So, I already know that Seurat puts all the cells together without
Suitable.
taking account of the biological replicates. So that's first problem.
Yeah.
They don't even assume it's all in one bucket.
Yeah.
So I heard that pseudobulk methods combined could be beneficial. What is
That's what industry does. So industry standard currently is to pseudobulk.
pseudobulk? But there are many pseudobulk. Like once you get raw counts for your cells, let's say this.
And you have to be very careful what population you're calling the same when you bulk, right?
Yeah.
When you put it together.
So then is there any programs that were developed over the years for bulk RNA-seq? Is there one that is better at dealing with that kind of distribution?
With the negative binomial?
Or when you have the counts, then that is gone and you kind of move to the space of a.
So this is the counts. So the negative binomial counts is so the cell by gene matrix that you get out and you're operating on.
But then when you put all the counts
when you
together,
pseudobulk them,
yeah,
right?
do you move the distribution?
Yes, then you move to this.
Okay. Okay.
Does anyone know why you can do that? This is a math. This isn't a fair question. This is a math question. It's the law of large numbers and the central limit theorem. It's because when you have a population, the average of a population, right? Pseudobulking is you take your single cells and you put them together in a population, right? So you've created a fake population. When you have a population, the average of that population, the mean, will trend to a normal distribution. That's why normal distributions are used so often and why they're so valuable. And once you get above 30 objects in the population, that's when that law kicks in really hard.
So then,
but those are 30 mice. Those are not 30 cells.
Yes. So basically, we never get to that.
No,
right?
it's,
So
well,
I
you
just,
can, but.
I
yeah.
played with pseudobulk just
Yeah.
to see, and I ran, say, DSeq, EdgeR, EBSeq, just this.
Yeah.
They all give different results.
Yes, they will.
So I know that you cannot give me the answer in general, but like.
I would go with DSeq because it's more conservative.
Oh, yeah. But so
and
there's no inherent program assumptions that work best for pseudobulk derived from single cell. Basically
Yeah.
based on the distribution you get, and also knowing that we rarely do more than 3-4 mice. That's already a lot.
Yeah, I mean, the thing is, so all of those are going to treat the multiple cells as the multiple independent,
Mhm.
so you just have to be careful. Yeah, I would do DSeq's the most conservative. I would, even if you're at 3 mice, do 3, n equals 3, and I mean, that's the reason that industry can do it, because they throw money at these problems for 30 mice isn't. But it's also the statistically most sound, right?
DSeq.
Oh, no,
30
the
mice.
soup. But yeah, no, DSeq's pretty good. It's highly it's more conservative. The other thing that you can do is do multiple algorithms and take the consensus. They'll have overlap in terms
Like
of.
Athg.
I mean, especially if you've already run it, so you know they got different answers. Just
Yeah,
see what
yeah.
everyone agrees on. Okay, 5 minutes. There you go.
Well, thank you for
I always feel bad. Why? Why do you feel bad? Because it's important, but also it's a little.
Burn up for what? We haven't done them yet.
Sorry.
Oh, I don't think he wants to work for that. Basically for Don, that's what has to go through and figure out, he uses very complex papers.
Hi, I just have a question about the discussion schedule. I'm like all out of sorts compared to when I think I'm coming. It's supposed to be like this.
Oh, you'll still be nice. Yeah.
Okay, for Friday. Okay.
So whoever is supposed to go today will have to, we'll figure that out.
Okay,
Everyone else is.
Okay.
And just the folks who will present it today.
Okay, and should I expect those papers to be posted over the weekend or will that?
Well, we're still, for
next Friday?
Yeah, we're still waiting on.
Okay, all right.
For each month.
Thank you.
And I was like,
Hi.
there was one, there was some kind of outlier that you guys have to solve. So I think I should be on higher once.
Oh, we're gonna report that.
Defaults are defaults. If your data perfectly fits that thing.
But I was like, why? I was kind of.
I just don't want to get
I mean, it's gonna be recorded, so you'll at least get the. So Gina,
What have we been doing?
we are going to figure out the discussion schedule.
Don't beat yourself
It
up.
will
You're right.
probably be the same paper that you did, but like we'll get the date and stuff finalized. So
Yeah, we'll have
to adjust only the date,
one with,
because I know you
actually the
put
paper that they're
the stuff
reading.
together, but it'll probably be the same thing. So
We had a sign reversal and somebody did a review on it. So like literally.
if that Wednesday is still open, then that solves. Yeah, because then she can just present that paper. Although, why would? Oh wait, that's right. Because she had a conflict with you. She couldn't make that day. Yeah, so it's definitely.